Our next presenter is Muthu Packirisamy. Muthu Packirisamy, Concordia Research Chair and Director of the Micro-Nano-Bio-Integration-Center at Concordia University, is a world leader on multidisciplinary area of micro-nano-integrated lab-on-chips for applications in life sciences. As a highly accomplished researcher, inventor, and prolific scholar, his technological breakthroughs and experimental innovations are used worldwide. Outstanding examples include intra-cellular-plasmonics and nano-interactions-based early cancer diagnosis.
Thank you for the introduction, and thank you for attending my talk today. So my talk is going to be on a convergence through direct sound printing and micro-nano integration is in a response to the call by Nobel laureate Philip Sharp on third revolution life science can tackle the 5 great challenges, grand challenges he mentioned, like you see brain or cardiovascular cancer diagnosis, discoveries, and education can be handled only through convergence. So my job is all about convergence. How do I work with people of different disciplinary? And so I do convergence working with collaboration through various engineering and life scientists, clinicians, through area I call it micro-bridging. So my area is micro-bridging, so connect people of different disciplines through micro-nanotechnologies. That's basically what I do. So why I choose this? Because in micro-bridging, there is a fundamental synergy, scaling synergy between your microsystems, whether you're dealing with from molecules to cellular level, from nanometer to micron level, and we make devices in the same range, from nanometer to microns and centimeters. There is a scaling synergy. Also, the fabrication techniques and measurement techniques, experimentation, all are common here. So I see there's a direct synergy, so I can get a high proximity to those tiny biological things by tiny devices. That's basically the idea why I chose this. So how do we implement it? I've developed 2 technologies here called lab-on-a-chip technology. Lab-on-a-chip is nothing but where you miniaturize all the functionals of a lab into a tiny chip. So basically replace a lab in this tiny chip of few centimeters in size. That we call the lab-on-a-chip. So that can cover anywhere from hundreds of nanometers to centimeters sizes. But also micro-nano integration is further miniaturizing. We can go up to the nanometer level by integrating these nano features and nanostructures into those lab-on-a-chips. So my area is all about that. So first thing is the direct sound printing. I'll give introduction to the direct sound printing. This is one of our recent breakthrough work on 3D printing with the sound waves. So far, the 3D printing has been done with either light or heat. All that you know. So first time we print with the sound waves. This is ultrasonic sound waves. Most of you might be doctors here already using HIFU for ultrasonic sound for various applications. We all use for medical applications. But here what I try to do is use this high frequency, high intensity ultrasound for printing applications. So what you do is the high frequency sound waves create a lot of cavitation bubbles. Through these cavitation bubbles, we create ultrasonic chemistry, which is used to polymerize materials to make from liquid to solid phase. So you can see the video here, that video that shows that how the polymer is being printed with high intensity frequency sound waves in this case. So we could print various structures. As you see here, a lot of structures has been printed. The machine is what you see is being printing those complicated structures we could do. And the right side, what you see here is where our technology is placed with respect to all existing present 3D printing technologies. And other beauty of our being ultrasound, we all know that it can pass through opaque bodies. It can pass through skin. It can pass through any opaque. So presently, the present 3D printing cannot print beyond the opaque bodies. But here with my ultrasonic height, we call the direct sound printing, I could print inside the body. This is what I'm doing called the remote distance printing that we do. So we can call it remote in-body printing in this case. As you see here, we printed behind porcupine, pig skin, and we can do complicated structures behind the pig skin also. So this is what you see here. We printed behind the right side, you can see the skin of the pig, and behind, we printed behind it. So one can extend this technology to print beyond skin and inside the body. This is my research that right now we are doing, is how to do in-body printing with minimally invasive surgery. This is what we are doing. I'm trying to expand into the bioprinting and organ printing. That's the kind of research that's happening here. So the whole idea is convergence. Again, I don't want to miss the point. I want to apply my technologies to work with various people to converge into real applications. So then another area of convergence is lab on a chip, as I told you before. So I do create a lab on a chip. So you can see here, the right side is that they are microfluidic chips, tiny chips. We can trap cells, we can culture them, we can manipulate them, we can do all kind of testing inside. We can create a different environment for the cells to grow. We can see various effects of environment on them, whatever the environment you are interested in. So we can basically pick each cell, name it, and then you can use it inside. That's the beauty of this lab on a chip concept. So we applied it on both the plant cells as well as human cells. A lot of work has been done on this area. I'll just give you one example here. We did on a pollen tubes, initially started with basically morphogenesis of pollen tubes have been done in this work. We try to understand what is the target pressure inside, how the pollens move, what are the cues that pollens respond to. So we study the effect of different cues here, whether let it be a chemical cue to steer those pollens when they want to search for ovule, what happens there, or it's a mechanical cue. All that we try to do on how pollens behave, actually. This is what. So we could do everything on a chip, and it gave a lot of interesting results about this morphogenesis of the pollen tubes. Then also we did work on energy harvesting from algae, basically blue-green algae. So here, when the blue-green algae undergo photosynthesis or respirations in the daytime, in the nighttime, in the redox reaction that happens, there is for every photon equivalent of that, there's the electrons are created. So what I'm trying to do here is, I'm trying to siphon those electrons and create electricity out of that. So this is what I did in this chip. So you can see the microfluidic chip. We have the proton exchange membrane, which is being used. On the right side, you see that energy created, power created with that. So we try to array this into multiples, stack them into multiples of this. The cells, I want to do something duplicate of photovoltaic cell, that solar cell. So I'm going to make a algae panel instead of a solar panel. This is what I'm going to do, go very green. We can see that we try to light LEDs bulb with that. Algae are working for us, but we can scale it up further for very serious applications. Then, early diagnosis of cancer. This is another work I do again on convergence side. So we have been working with very clinicians, doctors on various cancer detections, basically early cancer detection here. And I was basically based on detecting the extracellular vesicles, I call it exosomes in this case. So every living cells keep creating these exosomes, creating the bio-nanoparticles. These bio-nanoparticles have lots of information about the parent cell, whether it's in terms of mRNA, DNA, protein, fats, all it contains. So if we can detect those exosomes and understand what is content of those exosomes, then you can usually understand many things that go on. It's now exosomes are the new area where it's being used for diagnosis of various ailments, not only cancer. It has other things also could be done. Interesting thing that is, in terms of our early cancer detections, exosomes are much more powerful than circulating tumor cells. Okay, that's something which we found out. So we can go as early as possible here. You can see the curve that you show here, the curve that covers from no cancer to full cancer development here in this case. So basically what I'm trying to do here is just the concentration of those exosomes can tell you what stage of progression of cancer one is. So this is what I try to do, but this needs a very sensitive platform development. So we did a development of nano-integrated lab on a chip-based technologies for this. You can see one can predict the progression of cancer in this. This is sort of a liquid biopsy. It's very suitable for liquid biopsy techniques. I think this is what we are developing a lot into the liquid biopsy concepts right now. So on the other field of nano-bio-interactions, I was studying what's the effect of these nanoparticles onto the cells. So again, for a lot of most Indian ancient nanoparticles are being used for therapeutic applications. They call sonobashmas and various bashmas. Basically, they are gold nanoparticles and silver nanoparticles, all kind of nanoparticles. But they have been used in therapeutic aspects of cancer. So I'm trying to understand what are the fundamentals behind it, how these nanoparticles can interact with the cells, how they go into the nucleus, what kind of interactions they do. So I developed various methods based on one of them is basically the localized plasmonics techniques, the hyperspectral microscopy. Here we can see as the nanoparticles enter into the various organelles of the cells, you can see the plasmonic shift of those nanoparticles changes. From the plasmonic shift, one can identify what's happening around that, and it's up to now various possibilities opens up from there. Then also we understood the mechanobiology of impact on mechanobiology of cells with the nanoparticles, how they affect the mechanical motions and the strength of the cells. So mechanobiology effect one, you can see that how these nanoparticles can, for the cells that move, that we can understand what happens to the mechanobiology of the cells. And so this is my aim of my research is we try to miniaturize multifunctionalities of the entire lab into a tiny chip. This is what I do. It's basically shrinking an elephant into a bee with all the functionalities and strength of an elephant. This is what I do. And finally, I can take this technology to hospital to home kind of technology. So we can put all the functionalities of hospitals into someone's pocket so that it be useful, especially Canadian context. It's very useful for remote areas with well less access to the hospitals. Okay, this is what my futuristic work, and thank my team. Thank you.